for the honor of introducing our guest speaker today, I will turn things over to Dr. Leslie Walker Harding. Thanks so much, everyone, for being here.
Great. Good morning, and thank you, Suzanne. I am happy again to introduce our guest speaker today, who is one of our newer professors in the Department of Pediatrics. She joined the SCRI Research Institute and the Center for Global Infectious Disease Research in 2019. And we were really fortunate to have her come. And I'll tell you before I give you a little bit about her, she has a passion statement in one of her bios that I think really just typifies the joy it's been to have her with us. She says, for over 20 years, I have committed myself to 4 key scientific themes: discovery, translational research, global health, education, and mentorship. And she really lives all of those. And so I'm really happy she's here with us today. She received her PhD from the University of Washington and her MS from the London School of Hygiene and Tropical Medicine, and her undergraduate degree from McGill University. Dr. Kohler has over 20 years of experience in studying the pathogenesis of infectious disease pathogens, biomarker discovery, and diagnostics development in academic, biotechnology, and nonprofit environments. Her expertise in infectious disease and emerging epidemics was obtained largely through her roles as a student, technician, fellow, or scientist with positions held in the UK, the Caribbean, Africa, and the US. Dr. Kohler serves as a scientific advisor or committee member for global health and other epidemic-prone infectious disease entities, including the World Health Organization, University of Washington, Harvard University, and the Bill and Melinda Gates Medical Research Institute. She is also an affiliate professor in the Department of Global Health. And some of the things that she was able to quickly pivot to with COVID-19 is really looking at SARS-CoV-2 in kids and mRNA vaccine delivery. And so it's pretty exciting that she has some time to talk with us today about vaccine development. Welcome, Ria, and thank you so much.
Thank you, Dr. Walker Harding, and thanks for such a lovely introduction. I'm truly delighted and honored to be here talking with you all today for Grand Rounds. And I only wish we were meeting in person, but hopefully before too long, we will be able to meet again in person. As Dr. Walker Harding mentioned, I joined Seattle Children's Research Institute in December of 2019, and I really want to thank the leadership for providing a safe harbor for my team and myself. It has been an amazing ride since then. So I just wanted to, this slide sort of gives an overview of the topics that I'm hoping to cover today. I'll start with a snapshot of our TB vaccine development program, basically from the lab bench all the way to phase 2 clinical trials. And it's basically 20 plus years of work summarized in the following slides in the next 30 minutes or so. And then I'll start with some background studies and the rationale for the selection of a TB vaccine candidate. And this will be followed by a summary of some of the ongoing COVID-19 SARS-CoV-2 work that's ongoing in my lab. So I wanted to start first with highlighting my lab members. There are 15 of us, and these are really the amazing researchers who are getting it all done day to day. We focus on vaccine development and host-directed therapies against a variety of epidemic and pandemic diseases, and our mission is really to find solutions to combat tuberculosis. We have a small program in non-tuberculous mycobacteria and pandemic diseases. And really our focus is translational research. So we funnel vaccine candidates from the lab to human clinical trials, and then we characterize the immune responses of vaccines in preclinical and clinical studies. I'm an investigator in NIAID's Infectious Disease Clinical Research Consortium, the IDCRC, and I'm also along with the Kaiser Permanente Vaccine Treatment Evaluation Unit. We're a site that is responsible for blood processing and clinical immunology, primary endpoints and endpoint exploratory assays, including the evaluation of Moderna's messenger RNA 1273, which we were involved in the phase 1 clinical trial. We were the very first site here at SCRI and KP, and now we're involved in several booster studies that you've been hearing about in the news. So I'm going to pivot now to our work in Mycobacterium tuberculosis. This is Mycobacterium tuberculosis is a pathogenic bacterial species. It's the causative agent of tuberculosis, and TB is really believed to have been present in humans for thousands of years. It's been found in the prehistoric humans dating back to 3000 and 4000 BC. And it was first discovered in 1882 by Robert Koch. It has one of those unusual waxy coatings on its cell surface, and this is what makes the cells impervious to Gram staining, for example. And the physiology of Mycobacterium tuberculosis is really highly aerobic. It requires high levels of oxygen. And so for that reason, it's primarily a pathogen of the mammalian respiratory system. So Mycobacterium tuberculosis primarily infects the lungs, and it's most frequently diagnosed by a tuberculin skin test, quantiferin blood-based test, acid fast stains, and then chest x-rays. So it's been about 25 years since TB was declared a global emergency by the World Health Organization. And since then, there have been at least 10 declarations stressing the burden of TB and the need to develop new products to prevent, diagnose, and cure TB. But despite this, progress in the fight against the disease has been slow, with tens of millions of lives lost and hundreds of millions of patients, children, and families suffering needlessly. So in 2020, there were about 10 million people who developed tuberculosis, and about 1.4 million people died. 90% of those were adults, and 56% of those were people who lived in 5 countries. So India, Indonesia, China, the Philippines, and Pakistan. And so you can see from this world map the distribution of cases in the world. And mathematical modeling suggests that a vaccine modulating the natural history of infection among people who are already infected, latently infected with Mycobacterium tuberculosis, will play the greatest role in curbing the epidemic. So that's where we come in terms of trying to find solutions for this disease. It's basically responsible for about 5% of all worldwide deaths, and it kills more women worldwide than all causes of maternal mortality. And statistics show that approximately 50% of people who are infected with Mycobacterium tuberculosis and untreated, 50% of those untreated cases will die of TB. So a number of factors support the technical feasibility of a tuberculosis vaccine, including the fact that 90% of people who get infected with this pathogen do not progress to active disease. So the evidence really suggests that the infection itself provides some protection against new infections, and there is also a vaccine, a century-old vaccine called BCG, that provides partial protection in children. So, just given the complexity of infection and disease, there are four potential indications identified as targets for TB vaccine development. And these include prevention of infection as a target, prevention of disease, and immunotherapy, in which you can sort of give drugs or give the vaccine as an adjunctive to treatment with antibiotics. So in this context as well, we might give the vaccine as as a treatment to shorten regimens or increase the efficacy of the treatment of drug-resistant strains, in which there are many. And in fact, drug-resistant TB accounts for a little bit less than 500,000 deaths per year. So in 2018, there was a publication in the New England Journal of Medicine, well, a couple of publications demonstrating that a vaccine that I co-invented prevented disease progression in 50% of those who were infected with Mycobacterium tuberculosis. And this was really big news for the field. It was the first defined vaccine in 100 years that had shown, such promising results. And this trial involved some 3,500 participants in 11 centers in South Africa, Kenya, and Zambia. And really, it showed that the incidence of clinical TB was significantly lower in the group that received this candidate vaccine, this M72 and AS01E vaccine, than those who were in the placebo group. And so it's thought that that the vaccine might likely be effective if administered to young adults. And modeling has also suggested that a vaccine that's targeted to adolescents and adults would have the most rapid and cost-effective outcome on the global epidemic. I wanted to share this slide with you just to give you a sense of the funding and a timeline for development of this TB vaccine. As you know, it took us less than a year from having the sequence of the SARS-CoV-2 to rolling out the messenger RNA vaccines in December of last year. And here it is, you can see that it took us literally 30 years from the discovery of antigens, the development of adjuvants, and so on, before we started human clinical trials in 2003. And it was 2018 before we could demonstrate that the vaccine was effective in clinical trials. So M72AS01E was the first vaccine that we developed, and we went back to the drawing board and developed a next generation vaccine called ID93 and GLA-SC. The thought being that this antigen covers more phases of infection and disease, as well as incorporating a latency antigen. So the ID93 fusion, it consists of 4 antigens with diverse roles, and some of these are involved in virulence. They're involved in the PPE, so-called family of proteins, and it's combined with this adjuvant, which is a synthetic TLR4 agonist that's formulated in a squalene oil and water nanoemulsion. And studies, both preclinical studies and clinical studies, have shown that this vaccine is safe. It induces a T helper 1 type immune response, and it's scalable for local production. So I guess one thing that I should mention is that in our studies, we wanted to make certain that that we performed studies in mice, guinea pigs, non-human primates before moving into human clinical trials. And what I'm showing you here is that in mice, this vaccine induces a multifunctional CD4 T cell response. And we demonstrated that these responses are durable. They're responses that are seen in the spleens and the lungs of immunized mice. We also observed that the vaccine is protective against a challenge with Mycobacterium tuberculosis in mice again. So in this study here, you can see that the vaccine, the ID93 and GLA-SE, reduces the lung burden of the bacterial pathogens, and that the level of protection is very similar to the level of protection that is induced by the BCG vaccine that is used. We also showed that the vaccine in this mouse model is protective after 1, 2, or 3 immunizations. And the vaccine also protects against clinical drug-resistant isolates of Mycobacterium tuberculosis, whereas the controls, the placebo control or the adjuvant-only control do not induce protection. So just based on the body of data that we collected, we then moved on to process development of the vaccine candidates and then GMP manufacture. And what I'm showing you here is the clinical development plan involving phase 1, phase 2, and phase 3 studies. And the studies in blue that you see here are those that have already been completed. So we have completed phase 1 studies in the US and in South Africa, as well as a phase 2 study also in South Africa. And phase 2A studies have been completed in South Korea. And some of these studies have been done in individuals who are naive to Mycobacterium tuberculosis and have not received the BCG vaccine, whereas others, for example, in South Africa and South Korea, the individuals were immunized with BCG in childhood, as well as some of those individuals have been infected, and the thought was to just make certain that the vaccine is safe in those individuals. And so what you will see here are some of the studies that are planned. We're planning a phase 2B study in South Korea, Indonesia, the Philippines, and Russia. And there's also a discussion of a phase 2, phase 3 study involving 3,000 to 4,000 individuals. So we're currently working on the protocols and meeting with regulatory agencies and so on in order to get those studies approved for execution. So as you can imagine, there's been a lot of work and a few publications resulting from this work. And I'm showing you here just a subset of some of the individuals that have been involved around the world in bringing this candidate vaccine to human clinical trials. And when the M72 results were published, Barry Bloom had a commentary in the New England Journal Medicine, and he suggested that it might be that the adjuvant was critical, which we know, and also that the efficacy of the vaccine could be increased by the inclusion of additional Mycobacterium tuberculosis antigens. And so that is some of the work that we're doing in the context of our R01 and Impact TV contract funding. So evaluating the current vaccine and hoping to improve upon that vaccine with the discovery of new antigens and new delivery systems. So I'm just going to give you a snapshot of some of the results from the phase 1 and phase 2 clinical trials. And we did dose ranging studies evaluating different doses of the antigen and the adjuvant. We showed, for example, that a lower antigen concentration was better than higher concentrations. And we also showed that there was an induction of IgG1 and IgG3, suggesting that the vaccine induces a T helper 1 type immune profile. And in the evaluation of T cell responses, we saw high frequencies of T helper 1 type cells in individuals who were infected, and that the vaccine actually boosts responses induced by that infection. Now, this slide shows the robust polyfunctional CD4 T cell responses. And this study was actually done in individuals who were not just infected, but who had active disease, who had been treated with a combination of antibiotics. And on the left here, you will see the median cytokine positive CD4 T cell response, specific for this ID93 protein in whole blood. And it's stratified according to the different cohorts. So whether individuals received the lower dose of antigen and adjuvant or a higher dose of antigen, and whether as well individuals receive 2 versus 3 immunizations. So it's showing the phenotypic profiles of antigen-specific CD4 T cell responses in stimulated PBMCs on the right. What you're seeing here is basically stacked compass heat maps displaying individual CD4 T cell responses at 2 weeks post the last immunization of the vaccine. And so the columns correspond to the different cellular subsets evaluated, and they're color coded. And so white basically means off, and the shaded blocks means on. And they're grouped by the number of co-expressing markers in order of increasing polyfunctionality. So I'm just showing you like a brief snapshot of some of the data. And so you can see here that we're definitely seeing on the right here more polyfunctional responses post the second immunization. So we also have a couple of studies that are planned, as I mentioned. One of those is a prevention of infection study involving 180 healthcare workers in South Korea. And those were immunized with BCG, but were not quantiferon positive. And we're tracking individuals who received the vaccine versus those who are not, and then comparing the number of quantiferon conversions that we see in the vaccinated group versus the controls. And we've also followed that with an age de-escalation study in healthy adolescents. And we're basically doing those studies to plan for this phase 2-3 study involving 4,500 people in South Korea, the Philippines, Indonesia, and Russia. And so lots of plans, lots of work in the process. And so basically what I've shared with you so far is that the vaccine, this candidate vaccine is safe, it's immunogenic, it's versatile. And we're also, we continue to look for ways to improve vaccine development against Mycobacterium tuberculosis with lots of new candidate vaccine antigens and new delivery systems, including an RNA vaccine system that we are collaborating with a group in Seattle, HDT. And so stay tuned for what we can do next. So the next thing I'd like to do is just give you an inside look at the COVID vaccine development work that we have done here in my lab and at Seattle Children's Research Institute. So I borrowed the slide from Brittany Williams, one of the grad students in my lab, and it just basically shows, over time how the SARS-CoV-2 emerged. And so coronaviruses are a highly diverse family of a positive sense single-stranded RNA viruses. They infect humans and other mammals and avian species, including livestock and companion animals. And therefore, they're not only a challenge for public health, but also veterinary health and economic concerns, as we've seen. So these human coronaviruses have been known to circulate in the population, and they, together with the more recently identified isolates, cause seasonal and usually mild respiratory tract infections with symptoms that are really quite similar to the common cold. But in contrast, the severe acute respiratory coronavirus, so the SARS-CoV and MERS and SARS-CoV-2 have emerged in the human population over the past 20 years, and those are highly pathogenic. They infect bronchial epithelial cells, pneumocytes, and the upper respiratory tract in humans. And these infections can develop into severe life-threatening respiratory pathologies and lung injuries, for which, we don't have FDA-approved therapeutic treatments or prophylactic treatments to date. So the initial steps of the SARS-CoV-2 involves the binding of the coronavirus spike protein to the cellular entry receptors, which have been identified for several coronaviruses actually, and these include the human ACE2 that has been involved in SARS-CoV-2. So the pathogenic SARS-CoV-2 has rapidly spread in the human population after a likely spillover from bats or from perhaps a yet unidentified intermediate host. And as of yesterday, there were more than 209 million COVID-19 cases that were declared in over 200 countries worldwide. And that has caused more than 4.38 million deaths, again, worldwide. So I wanted to share the phase 1 trial timeline. My team has conducted many vaccine studies over the years, but the task before us in March of 2020 was really different. The timing was compressed and the stakes were higher. As I mentioned, we had moved to SCRI just 2 months prior, And purely by coincidence, I was at the NIH when the CDC confirmed the first case of COVID-19 in Washington State in January. And that involves, someone who had recently returned from Wuhan, China. And so, we frantically started working on the clinical trial protocol. And then, along with Kaiser Permanente, the group led by Lisa Jackson, my group was then chosen as the first team that launched the first US human clinical trial for this Moderna messenger RNA vaccine, which presented hope. So it's amazing that just 75 days after the news of these unexplained cases of pneumonia emanating from a seafood market in Wuhan, China, and just 66 days after scientists in the US were, given the virus's genetic code, that we started here at SCRI receiving the first blood samples. And so now we are, currently implementing additional trials evaluating other COVID-19 vaccines in the pipeline, and evaluating the efficacy for mutated or the so-called variant forms of SARS-CoV-2 that are popping up around the globe. And I just want to, have a shout out for not just my team, but also everybody in our center business office, EH&S, the office's sponsored research, IT, HR, and of course leadership, because honestly, all of these groups were so integral to the progress that we made to to the accomplishments to date in my lab. And really developing a safe and effective vaccine involves such a massive collaboration, not just among scientists and their support systems, but also, as you can see, from what we've been able to do in less than a year, it really requires collaboration among government organizations, researchers, pharmaceutical and biotechnology companies, and of course funders. So I would say that we were really thrilled to use our experience and our capabilities in clinical immunology to assist in these efforts. So now what I'd like to do is just give you a brief overview of some of the clinical immunology that we undertake in my lab. We have worked on developing various clinical protocols for the vaccines that we are evaluating, either at the IDCRC or the VTU, or through funding of biotechnology companies locally and internationally. So we do everything from reviewing the draft informed consent forms, updating what we call an investigator's brochure, evaluating the pharmacy plan, and looking at, and preparing the clinical study reports at the end of the study. So we're involved in everything from the clinical trial preparation, so the sample management, specimen processing, site training, and then obviously also involved in some of the clinical immune assay assessments, and that includes humoral assessments, cellular neutralization assays, pseudo-neutralization assays, intracellular cytokine staining, and we execute what we call qualified or validated assays through really extensive laboratory testing. So we're looking at the performance characteristics in mathematical and quantifiable terms for ELISAs, ELISpots, ICS. And these assays are really based on predetermined acceptance criteria. And it's really to demonstrate that the assays that we're using to evaluate the immunogenicity of the candidate vaccines are suitable and that they're reliable. And so we qualify we validate the assays for what we call linearity, precision. We look at the limits of detection. We make certain that the assays are specific and that they are robust. So taking these skill sets, we pivoted very quickly into helping with the execution of the Moderna messenger RNA 1273 evaluation in the clinical trials. And we've been involved in the original vaccine that was developed by Moderna, as well as follow-up vaccines that have been developed to address the variants that have been popping up around the world. So, that initial trial involved 45 adult participants, 18 to 55 years of age, and then and those individuals received different doses of the vaccine, 25, 100, or 250 micrograms. And then this was quickly followed by a phase 2 and phase 3 trials of a 2-dose schedule of the vaccine given 28 days apart in adults. And there have been several publications that have resulted from this work, of which my team and I were involved. And so the safety data were gathered after the second dose in the trials, and we saw that the vaccine was safe, that the side effects included the typical headache, fever, some muscle aches, but there were no serious adverse events. And so, many people have asked, well, how is it that the vaccine development timeline was so accelerated, and how do you do that at an accelerated rate while, still ensuring that it's safe? And I guess, there are a couple of points, to mention. The RNA vaccine technology had been around for, had been in development for 20 years as well. And of course, these vaccines are much faster to produce than traditional vaccines. And I think also another important point was that when this was rolled out, the researchers used existing clinical trial networks to evaluate the vaccines that were in development, which I think really helped to accelerate. So binding antibody IgG geometric mean titers to the spike protein were evaluated. The study showed that there was an increase after the first vaccination with seroconversion in all participants by day 15, and that there was a dose response seen to the first and second immunizations. And we also saw pseudo-neutralization responses were detected in, I guess, less than half of the participants, and a dose effect was also seen. But after the second immunization, pseudo-neutralization responses were observed in the serum samples from all participants. And because we saw that the responses were similar in the 100 microgram dose and the 250 microgram doses that were evaluated, we then, went with the lower dose. So we also saw that the vaccine elicited CD4 T cell responses that on stimulation by the spike specific peptide pools, they were strongly biased towards T helper 1 cytokines, so TNF, interleukin 2, and gamma interferon, with minimal T helper 2 expression of IL-4 and IL-13. And interestingly, the CD8 T cell responses to the spike were detected at low levels, very low levels, after the second vaccination in the 100 microgram dose group. So we also looked at the safety and immunogenicity of the vaccine in older participants, and again we saw, that the data were really quite good, and also looked at the durability of the responses in individuals 119 days after vaccination. And again, we saw that there were high levels of binding and neutralizing antibodies that declined only slightly over time, but they remained elevated in all of the participants months and months after the booster immunizations. So the phase 3 demonstrated 94% efficacy against symptomatic confirmed COVID-19 cases 14 or days or later after the second vaccination. And then 100% efficacy was seen against severe COVID-19. So as you know, on December 18th of 2020, the FDA issued an emergency use authorization of the vaccine, and the vaccine was rolled out to adults in the US. So recently, SARS-CoV-2 variants with mutations of the spike protein have emerged, and this was first identified in South Africa, and so we are involved in vaccination strategies that hopefully induce broader protection that includes variants such as the South African variant, as well as the more recent Delta variant. And so we're involved in many additional clinical trials evaluating these variants and different boosting protocols. And just want to give you now like a snapshot of some of the additional clinical trials that we have been involved in, not just with Moderna, but with other groups. So we're actually collaborating with the Institute for Protein Design, Gridstone, and the IDCRC to evaluate this chimpanzee adenovirus self-replicating messenger RNA prime boost protocol. And we're looking at responses in adults, so 18 to 60 years of age, and really focusing on heterologous and homologous protocols. And so our role in this study is to evaluate the immunogenicity by evaluating total IgG against the Wuhan spike, the Wuhan receptor binding domain, as well as variant spike antigens. We are also collaborating with a group located here in Seattle, HDT, and involved in evaluation of their candidate RNA vaccine, which is a self-replicating replicon RNA vaccine, and that is delivered with a lipid inorganic nanoparticle, so a lion particle. And we again are involved in the endpoint ELISAs, as well as looking at pseudovirus neutralization and a live virus neutralization as endpoints. Okay, we're evaluating different vaccine doses and different delivery regimens. So it's really exciting to be involved in so many different studies. Another study that we more recently got involved in is one in which it's an observational study to determine the safety and immunogenicity of SARS-CoV-2 delivered during pregnancy or postpartum. And so the CDC has been tracking more than 30,000 vaccine recipients who were pregnant at the time of vaccination, and nearly 1,800 of those have provided detailed descriptions of symptoms after vaccination and pregnancy outcomes. And so, again, our role in this study will be to conduct a SARS-CoV-2 pseudo-neutralization assay to characterize neutralizing antibodies in individuals who were vaccinated while they were pregnant or postpartum, and then the transfer of immunity to their infants. And so this pseudo-neutralization assay will be used to quantify antibodies from the serum or plasma samples that are capable of neutralizing SARS-CoV-2 pseudovirus entry into and the ensuing replication within cells. So what I've shared with you today is that SARS-CoV-2 has caused, well, this is out of date because it's not 3.3 million deaths. It is 4.38 million deaths as of yesterday. And there have been 209 million cases, and we've had about 1.86 billion people worldwide who are fully vaccinated. So we have a long ways to go. And so the battle to stop the pandemic is not over, but hopefully we will be better prepared for the next one. And we continue to work on trying to understand the immune responses to SARS-CoV-2 infection, because really there's such a lack of knowledge about the immune responses that are needed, not just for infection, but to prevent severe disease, because we're seeing that the responses are different. We've shown that at population level, studies show that individuals who recover develop not low levels of neutralizing antibodies, indicating that neutralizing antibodies are likely to have a relatively limited impact on resolving disease. So the studies are showing that that these neutralizing antibodies may be more critical for protection against infection than for resolving disease. At any rate, there's a lot of work to be done. We are currently also adapting our assays to use breast milk samples for the infant and mom study that I mentioned to you, and we're just thrilled to be involved. So thank you. And this is a list of our funders and our collaborators, and you'll see that we're working with lots of people, both locally as well as internationally. And we thank the clinical trial participants and everyone who has helped to get us here. Thank you very much.
Thank you so much for all your work in this really, really important world. Everyone who's on the call right now, if you want to type questions or comments into the chat, or if you want to be brave and unmute yourself and ask a question, we have about 10 minutes for Q&A. Thank you so much.
Hi, Ria. This is Ellen. I had to unmute myself to tell you what a fabulous talk you just gave. Thank you so much. I am grappling a bit with a question that I did ask you a couple of days ago about patients who have had COVID and are resistant to getting the vaccine. One of the reasons being that they've heard that people have worse reactions to the vaccine if they've had COVID. And another one being that they are concerned about the myocarditis, pericarditis issue. And this issue has become more personal for me because my son just found out that one of his roommates that he's supposed to have starting college is not vaccinated because he had COVID, and my son is super uncomfortable with that. So I'd love your thoughts on this particular issue and the risk of being in close contact with someone who's unvaccinated because they have the disease.
Right. Thanks so much for joining us today, Ellen, and for your question. You know, I hear what you're saying, but really, we have not seen a causal link between uh, like antibody abundance and enhancement uh, of disease or enhancement of symptoms in individuals who have had the disease and who have been vaccinated. So I know that, because of what we've seen, in the context of dengue, for example, where you see this antibody dependent enhancement of, um, of symptoms, we have yet to observe that in humans who are infected with SARS-CoV-2 and who are then immunized, or who were infected and then immunized. The other thing that I would mention is that, and I sort of alluded to this when I was presenting some of the Moderna studies. What we've seen is that the neutralizing antibodies may be critical for protection against infection, but not necessarily important in resolving disease. And so there are lots of different things to think about, right? In terms of the types of antibodies that are induced with infection versus the flavor of antibodies that are induced upon vaccination. And so I think, some of the extra neutralizing functions that that are induced by vaccination, including, the ability to recruit antibody dependent cellular phagocytosis or complement dependent cytotoxicity and ADCC, the antibody dependent cellular cytotoxicity. These are some of the additional functionalities that are important in also resolving disease, so not just preventing infection. So, this is why I recommend that even individuals who have had COVID-19, that they should still be immunized. My personal belief.
Thank you so much. And do you feel there's a risk of being in close proximity with an unvaccinated person who's had the disease versus someone who's vaccinated?
I do, because again, those the people who've been infected over time, those antibody levels wane, right? We've clearly shown that. And we've even shown in our studies in individuals who have received the vaccine 8 months, in some cases, as quickly as 6 months after being immunized, we're seeing, breakthrough infections. And so the vaccine is still, 6 months out, 8 months out, preventing severe disease, but it's not preventing infection. So whilst we're seeing that, that there isn't an increase in hospitalizations, for example, in those individuals, they're still, experiencing symptoms from COVID-19.
Thank you so much.
Thank you, Ellen.
We still have time for a couple more questions if anybody's brave enough to talk or type in the chat. In the meantime, can I just ask you, this is a bigger question, but any predictions for what will happen as school is about to start in our area? And recommendations from you as we advocate to different leaders in this on the school leaders.
Right. Thank you for that question, Suzanne. I honestly think that everyone needs to be vaccinated, right? until the world is vaccinated, we will continue to see the emergence of additional variants. And that is what we need. We're seeing that there is a decrease in effectiveness in guarding against infection, while maintaining the ability to prevent severe hospitalizations and death. And when you look at the hospitalizations that are occurring, more than 95% of those are involving individuals who are unvaccinated. So even in kids, I would say, as soon as they can be immunized, they should be immunized, and we need to continue um, masking, social distancing, washing our hands. I mean, I'm sorry that we're back. It seems like we're back where we were a year ago, right? And it's depressing, but hopefully given that the US and Canada and other countries are sharing their vaccines with the rest of the world, I'm hoping that before too long, most of the world will be immunized, which will also prevent the emergence of other variants.
Agreed. Can't happen soon enough. Thank you. And
Hopefully.
from Dr. Stapleton, how important is getting a booster in someone who has had COVID and 2 Moderna or Pfizer vaccines?
Thank you for that question, Dr. Stapleton. I think it turns out it's important. We're seeing that in large scale studies that the efficacy in preventing hospitalizations is about 86%, and which is really quite good, right, in terms of preventing hospitalizations. But there have been other studies that have been shown that the efficacy for preventing hospitalizations, for example, in nursing homes, there are some studies that are showing that the efficacy is only about 53%. And we're definitely seeing a waning of responses 8 months after immunization. So I'm personally a proponent of the booster vaccine. And I'm not just trying to push vaccines because I'm involved in vaccine development.
I think many of us are counting down to when we are eligible to get it. So, agree. I think you have a lot of agreement in this group. All right. Well, it looks like it's almost 9 a.m. So I'll just pause for a second to see if anybody else has any final comments.
Yes, Ria, Dr. Kohler, this is Leslie. I just wanted to say thank you. This is really, it's so exciting to have you here doing this work and keeping us up to date on everything. So thank you very much.
Thank you so much, Dr. Walker Harding. Thank you for
Yes.
inviting me today.
Thank you so much. We really, really appreciate your work, and let's get get this done. Thank
Yeah.
you. Thanks, everybody. We'll see you all next week.